In the air all around us are countless microorganisms, bacteria and fungi that have become airborne as an air mass moves across the Earth. These can be collected using machines like the one you see here to try and understand the potentially important roles they play to terrestrial patterns of diversity, especially in sensitive and changing places like Antarctica. We've produced this video to share with you the science behind the global Aerosphere project and the wonderful journey involved in gaining this unique global dataset. We've had the privilege to investigate airborne microorganisms in various locations around the world, but have recently received funding to complete a comprehensive dataset in the locations you see here. The sampling includes a network of near-ground aerosol collections that will be supplemented with targeted high-altitude collections in helicopters and planes, removed from local influences and at a location that is believed to be the basis of longer-distance movements. We wanted to complete the targeted sampling with as little air travel as possible. To do this, we decided to separate the remaining sampling into two trips, one with each hemisphere of the Earth. The first trip took me around the southern hemisphere, starting from my home in New Zealand with a first stop in Perth. I was very fortunate to be put in touch with the owners of a farm a couple of hours outside of Perth thanks to my in-laws. It was very dry at the time of the year, but within a few short weeks there would be an explosion of life as the rain came. I set up 3 samplers to reduce error and increase material to work with in the minimum amount of time, and collected soil samples to compare to the overlying air to determine local influences. All 3 run at the same time towards the wind to reduce human contamination from its biggest source, me. In the laundry of the farmhouse, I set up a makeshift laboratory to process the samples, as I couldn't travel with the heavy, bulky items or bleach that I needed to clean the equipment between samples. I acquired these from local collaborators at each stop. Each sample was filtered using a 0.2 micron polycarbonate filter to catch the microorganisms. The filters would then be placed in a preloaded tube with DNA preservative buffer to be shipped back to New Zealand. In addition to the ground sampling, Perth was one of the locations I conducted high altitude sampling. A local company was able to remove the door for me so that there was a free flow of very cold air from that altitude rushing past the machine for collections. At this altitude there would be much less ground influence and the air can travel much greater distances. After a few short days of sampling in Perth, I was off to Pretoria in South Africa. The University of Pretoria allowed me to use their farm facility for sample collection, and once again I had set up a small work area to process the samples as cleanly as possible. As of course these aren't laboratory conditions, I had to make sure there were lots of controls to take into account any accidental contamination that may have occurred. I did actually manage to get a day off in Pretoria due to logistics preparations for the next stop, where I was able to go on a small safari only 15 minutes from where I was staying, which was an amazing experience. After Pretoria, a quick flight across to Walvis Bay, Namibia allowed me to meet up with my colleague and co-PI of the project, Professor Stephen Pointing from yale-NUS. In addition to collecting biological samples at each location, I would also take down the total particle loads of the air, as well as relative humidity, temperature and airspeed. These factors are believed to be highly important in determining what's in the air at any one location. At each location, each machine would collect 6 one-hour samples per day for 2 days between 10 AM and 4 PM. Once again, I set up my sample processing laboratory. Here you can see me cleaning everything inside and out with bleach to destroy the DNA from contamination. Bleach is wonderful to remove contamination, but you also need to be careful not to let it come in contact with your sample, or it will destroy that too. When we met our pilot in Namibia, we were pleasantly surprised to find another New Zealander who took great care of us. Namibia is an incredibly dry place, but still with a wonderfully diverse array of life, ranging from lizards, beetles, spiders, snakes, and many types of trees and shrubs. The desert varied in a surprising amount from desert pavement, which looked like gravel, to of course the beautiful sand dunes we all associate with the desert.
It's a sidewind.
After we completed our work in Namibia, I was off on my own again to Montevideo, Uruguay, where I met one of my collaborators who arranged for me to sample in a farmhouse 2 hours outside of Montevideo. The scenery in Uruguay looks surprisingly like back home in New Zealand. It's a beautiful and much cooler country with a lot more life and a welcome change from my previous stop. The farm I stayed at dated back to the 1860s and had a wonderfully accommodating host who even prepared an amazing traditional barbecue while I was there. The samples were packed up to be shipped back to New Zealand by my collaborator, and I was off to Chile, where I'd be sampling in the Atacama desert, and possibly the driest place on Earth. The air samplers were set up with absolutely no signs of life visible, other than myself and my collaborator. These tire tracks have been laid down by many sampling trips over at least the last 20 years. This is a very popular spot for NASA scientists trying to understand life pushed to its absolute limits in the hope of using this information to guide their search for life on other planets like the upcoming Mars mission. With only a small amount of difficulty, we managed to charter a helicopter and once again sample at 2,000 meters altitude, taking in the beautiful and barren landscape with only the occasional solar farm or mine to be seen. For this leg of the journey, I was joined by my NASA collaborator who I assisted with another project looking at the microbiology at different depths under the Earth. Now you see, rain doesn't happen often in the Atacama desert, but a few years ago there was a massive downpour, and to this day some areas under the Earth are still a little wet. We hope to see if this helps life survive for longer periods of time than that on the incredibly dry surface where there's no signs of visible life. The sampling had to be done with the utmost care to ensure that no human contamination would get into the samples that had so little life in them to start off with. The sunsets in the desert were absolutely mystifying, with a warm green glow reflecting off the ocean and red oranges reflecting off the dust particles in the air. Our southern hemisphere sampling completed, I was heading back to my family in Auckland to process samples before my next trip across the northern hemisphere. Science at this scale involves an incredible team with a broad range of specialties and access to special locations for science. This work would not have been possible without them and the many other citizen scientists and support crew who eagerly assisted me whenever I needed it. My team and I are honoured to be able to take another step in understanding this poorly understood ecosystem, to be able to work with so many amazing people, and to be able to share this knowledge with the general public. Thank you all for your attention, and stay curious.